So we want to open this with Excel. Here is what I have found is my most used program probably for what I do is Excel, hands down. And I've talked to many biostatisticians and computer people that like, oh, you can't use that because some kind of control, blah, blah, blah. Heck with that. It works, and it works really well. And everybody has a copy of Excel. So open this with Excel. You can kind of open up these columns. Can everybody see that? Do you want to make it bigger? Is it okay on the screen? Okay. So now you're going to see why I kind of wanted you to put the genes in the columns. Everything here is in rows. Okay? Here's what we're going to need to do is we're going to need to format this. Okay? Under source name, this is going to be our uniting identifier. We're going to take this file and based on this column, we're going to merge it to the file that we just downloaded. Okay? Here's the problem. Go back to your Partech. What does this file name say? Is it the same? No. Yeah, this one has text, and this one has a 1 at the end. Let's get rid of the text first. So, are you in Partech? Sorry, I keep moving back and forth. This is what you have to do. Under file name, what I want you to do is, let's see. Should be a find replace. Go find replace select. Under find what, I want you to put dot text. And replace with what, just don't put anything. And hit replace all. Oh, I did it in all columns. Dang it. Sorry. Is yours still going too? So it's going through 35,000 things. Tell me when it gets done. I mean, actually, I'm going to hit close. Let's do it again. I'm sorry. Hit close. So file, edit, find, replace, select. Only in column file name. Now I'm going to hit replace all. What? Should we replaced it? Column file name. No one else has worked, right? Mine's the only one that didn't work. Dot text. Replace with 0. Replace all. Oh, yes. Yay. Okay, so we got rid of that. Got to do the same thing on this other column. Again, edit, find, replace. So in this one, do a space and then 1. Okay, replace all. Should have only made 30. If it makes more than 30, that means it took some stuff out somewhere else. Yeah. It might have done it on the other side. I don't know. Hit undo. So you're on Excel. You can do undo. All right. Trust me, I have spent hours upon hours going, why isn't this thing merging? And then find like there's like some comma or something that I didn't add, or just drives me insane. Okay. Is everybody with me now? Yep, cool. All right, let's just go column to column. This is kind of a good one. We can kind of see what's going on here. This all says the same thing. We can get rid of that. So get rid of column B, go delete. Source name. This is a good column in that it gives you everything that's in that study, basically in the name. And whenever I name anything, I always put everything I can into the name, which drives people nuts. They're like, Mike, why is your file title 80 characters? And I'm like, because there's a lot of stuff in there. Here's what we want to do is we want to basically take some of this. So if we look here, everything kind of transfers, right? They've got the clones, they've got the characteristics of the cells. Here's the information that we don't have, is we don't have days, right? And if you read the study, so let's go into the study and talk about that for just a minute. Here's what they're doing, is they're basically taking embryonic stem cells or pluripotent stem cells from mice. They're basically using these to try to differentiate them so they can make endoderm. And if they can do that, they can treat diseases like cirrhosis of the liver, emphysema. Basically, you can make skin. And if you can make skin, you can do a lot of things with that. Okay? There's, again, in this study, there's 2 different ways to make skin. You can use embryonic stem cells, which are very hard to get and expensive, or you can do induced pluripotent stem cells, which are much more desirable because you can get them from adults. Okay? And basically what they're doing is they're trying to figure out which way works the best. And what they use is they use these markers. So FOXA2, and there's this, I think this cytokine receptor 4. Basically, as these cells differentiate, this signal gets stronger. So they're basically using these markers of endoderm development to basically take a sample at day 0 before they're induced. And then at day 5, when most of them show that they're differentiated. And they know that based on the markers that they use. So when we go into those files, you'll see sorted by blah, blah, blah. They're sorting by these markers here. And they're doing that so they can only get the differentiated cells. And this is basically this is how they get these things. They take the cells, they put them in mice, they breed them to a white mouse, and then it basically comes out and they can harvest them from the litter. Pretty cool, but barbaric. I'm glad I don't have to kill mice anymore. Anybody here killing mice? Yeah. Yeah. I used to dissect their brains. I worked in a lab. We were doing the aging brain. And so we'd have to dissect rat brains all the time. So I get a call home and go, "Hey, Mom, I'm a brain surgeon now." She's really? And I'm like, "Yeah." And she'd be like, "They're not letting you really on people, are you?" And I'm like, "No, they're not. It's okay." Okay, let's go back to so we kind of describe what's going on here. They've got some other things. Like I said, you could read half this book here and, figure out what they're doing. I read it, and I have a hard time understanding what's going on. But once you see the data, I think what you're going to find is it'll become pretty apparent. Here's what we need to do is we need to put some more columns in here, because I want to get the day. Right? Oh, just got to make sure my daughter's home. Cool. Okay. So let's insert a column here. Okay, we're going to make this day. Okay? This is how I do this. This is very technical, so pay attention. What I do is I put a 5, and I copy that, and then I just paste it wherever I see the 5 over on the other column. And I know, yes, you can do programs that do this for you. By the time you wrote that program, I'd be done. And then let's do it for the day 0. Same thing. There is a lot of cutting and pasting in bioinformatics. I'm just going to warn you right now. Okay. I'm going to expand this once I'm done. So some of these, they don't have anything. Let's put a... So I'm going to tell you right now, Partech does not like blank squares, right? It freaks it out. It's like, what's in there? So if there's nothing, so obviously, basically what they wanted to do is they did a bunch of a mouse embryonic stem cells or cells from the endoderm. They want to compare it to the differentiated state. So just put a, those don't really have a date, so put a question mark. Okay, there was one other thing I wanted to get out of this. Oh yeah. Let's put it in another category. Insert. Actually, here's what I want you to do. Highlight column B, copy it, put it right after C, and then right click and go insert copy cells. Okay. Let's do our find and replace again. So go to replace. Here I want to keep that sorted thing. So I want to find, actually, I want to find this. Actually, it's the semicolon. So I want to get rid of in a space. Replace with nothing, and then replace all. Oh man, I did it wrong. Sorry. Wait a minute. So replace, I think you need to put a star. So if I go star, replace with 0, in a space, replace all. Let me try that. Ah, it worked. Did everybody get that? It's okay, that's not going to be a huge column. Here, let me help. Did everybody else got that? Okay. A lot of like crunching beforehand before we can have fun and see visualizations and do crazy t-tests. This is you know, wax on, wax off kind of stuff, you know, Karate Kid. You know, it's, honestly, most of my time in bioinformatics is spent kind of formatting data to get ready to look at it. So, and I will say, like I said, Geo is horrible. And it's one of the worst data sets to get information out of. And I always, you know, I end up having to do it a lot. So I end up formatting a lot. Someday there will be another data set, and I will promote it. I'll do commercials for it. But you can see how, okay, wait, let me make sure we got everything sorted. Let's go organism here under musculus, even though it's the same one. This would be, let's go genotype under G. Here we want strain. And here we want tissue. And you know what to put because it was in that bracket thing. I just like seeing the column headers Oh, wait. And under here, let's just go under comment, sample source name. For that header, let's go all. Because it gives you all the information. All right. We now have an Excel file that contains all the experimental data for this study. What do you think we're going to have to do with this now? Import? No, we're not going to import it. We're going to paste it. Watch this. You're going to love this. You'll notice I get excited about data, which is, in my business, you have to. All right. You'll notice, like right here, this A, between A and 1, click in the middle there and it'll select everything. Hit Control C or copy or however you want to do it. You can do it through the menu. Now let's go back to our Partech. Under Edit, I am going to go Paste to a new spreadsheet. Yeah, 6 missing data. Okay, that's all right. Yeah, the question marks are still there. Oh yeah, 1, 2, 3, 4. Yeah, I guess you're right. Probably. But it didn't ask like what to put with it. All right. That was pretty easy, right? I mean, the part about getting into Partech, not formatting the data. Here is what we're going to do is we're going to merge these two files.